Hello everyone, and welcome to this podcast for genetics. And in this podcast, we're going to focus on the genetic control of cancer. Okay, as we get started, let's remind ourselves of a few features of the cell cycle. Because cancer is largely a disease of the cell cycle, and mainly the mitotic cell cycle. So we're going to be focusing on mitosis. Earlier in this unit, we focused on meiosis, but cancer, really, we have to think about mitosis. So just like meiosis, mitosis has the G1 and G2 with the S phase in between and the M phase up here. What we didn't talk about are these checkpoints, and we know there are a few checkpoints. And I'll explain why they're important in just a moment. The first one here is the G1S checkpoint. So let's draw a cell here. The cell is entering the cell cycle here to copy itself through mitosis. It stops at this G1S checkpoint to make sure that the cell is ready to move on to S phase. It checks for a few things. Is the cell the right size? Are the molecular components ready for S phase? Meaning the cell is getting ready to copy the genome. Are the enzymes necessary to copy the genome active here? And it will also check for mutations. If there are no mutations, then it's ready to copy the genome. If there are mutations, the cell will do one of two things. And these two things are either fix the problem, fix the mutation, turn the right genes on to enter S phase, wait till the cell is the right size. So it might fix the problem. However, sometimes the problems are at such an extent, it's not easy to fix, and in which case the cell will enter apoptosis. And for our purposes right now, just remember that apoptosis is programmed cell death. The best example of this is when you're out in the sun for too long and you get a sunburn. Your cells try to fix those mutations, but at some point they can't. And so the cells enter apoptosis, and that's why your skin peels. The skin is peeling because those are dead cells that cannot fix the problem. So we want apoptosis to occur in this case because we don't want those mutations to be passed on. Next, let's talk about the G2M checkpoint. At this point here, we have a similar list that the cell will go through. But let me actually draw our cell here. Our cell has moved up here, it's moved through the S phase, and it's copied the genome. And so it needs to determine at this checkpoint if the cell's the right size. Again, up here we're going to split the chromosomes into two new cells, so the cell needs to be the appropriate size. Again, it will ask, are the molecular components present to enter M phase? Is the genome completely replicated? Has all the DNA been copied? And of that DNA that's been copied, are there mutations? Again, if any of these problems exist, the cell will first check to see if it can fix those problems. But if it can't fix those problems, the cell will enter apoptosis and die. Our third checkpoint is up here, and it's called the Spindle attachment checkpoint. And the title of this checkpoint is pretty self-explanatory as to what it's doing. Remember, our cell is going to be here, and I'm not going to draw great detail here, but remember those spindles have to extend outward to attach to those chromosomes. And until they have properly attached to the chromosomes, the cell cannot move into anaphase. Can't complete metaphase and move into anaphase. So it'll stop here. Again, it either fixes it, it waits until they're attached, or the cell will die. Again, this is very important because if they don't attach properly, that's when we end up getting things like the aneuploidies or the polyploidies. So it's important that this checkpoint is followed closely. So as our cell is moving through the cell cycle here, it stops at each of these points to make sure that the cell is ready to proceed. And this works pretty well most of the time. But we are going to talk about cancer today. And cancer occurs largely due to a failure in one or more of these checkpoints. When these checkpoints aren't followed properly, you will pass on mutations to the next cell generation. Before we talk about mutations and what they do, let's talk about something called cyclins and cyclin-dependent kinases. In a cell biology course, we would go into much greater detail about cyclins and cyclin-dependent kinases, but I just want to touch on them here briefly. So cyclin-dependent kinases. If you remember from a previous course you've had here, you might remember that kinases are enzymes that phosphorylate other proteins. And in doing so, they will often trigger specific pathways. In this case, they're going to trigger specific pathways to move past these checkpoints. We often just call these CDKs, and so that's what I'm going to call them in this and other podcasts. A CDK by itself is inactive. And there are different CDKs that are important at each of these checkpoints. So as long as it's stopped at this checkpoint, it will remain an inactive CDK. But to be able to move from G1 to S, or from G2 to M, or past the spindle attachment checkpoint, a specific CDK has to become active. And it becomes active by adding this protein called cyclin. And cyclin is a protein that binds to CDK. So you'll have your CDK protein here, and it will bind cyclin. And now the CDK here is active. Cyclin-dependent kinase, meaning that it's a kinase that is dependent upon cyclin. Once active here, the CDK cyclin will phosphorylate specific targets. Once active here, it phosphorylates targets that allow it to progress into S phase. And same thing here. The CDK cyclin will phosphorylate specific targets to allow it to move into the M phase. Now, I'm going to start moving into talking about specific proteins that regulate this cell cycle and ultimately regulate whether or not cells will remain healthy or begin the multi-step process to becoming cancerous. Cancer-linked genes can be lumped into one of two major categories. They can either be called tumor suppressor genes, or they can be called proto-oncogenes. We'll talk about examples of each tumor suppressor genes and proto-oncogenes as we move forward. For now, I'm going to give a very brief description of each of these, and then I'm going to go into greater detail. You should think of tumor suppressor genes as genes that encode proteins that stop the cell cycle. Tumor suppressor genes are active here at this checkpoint, and this checkpoint. And what they are doing is stopping the cell cycle until it's the right size, the right proteins are being made, and once it's been verified there are no mutations. If you're thinking about a car, you can think of tumor suppressor genes as the brakes of that car. They stop the cell cycle. Now, proto-oncogenes are the opposite. They are the start process of the cell cycle. If you were going to use the car analogy where these are the brakes, these are the gas of the cell cycle. So in order to get past this checkpoint that's being held at a stop point by these tumor suppressor genes, the proto-oncogenes have to become active so that they can move the cell cycle forward in each of these cases. In order to properly regulate this cell cycle, there must be the proper regulation of tumor suppressor genes and the proper regulation of proto-oncogenes. When these become mutated in certain ways, that's when the cell cycle begins to grow out of control. Now, something else that is characteristic of these two classes of genes is that tumor suppressor genes typically behave as if they are recessive, and proto-oncogenes typically behave as if they are dominant. And I'll talk about this. The tumor suppressor genes, typically both copies of the tumor suppressor gene need to be mutated. With proto-oncogenes, typically only one copy is mutated. These mutations for tumor suppressor genes are typically non-functional. So what I mean is that when both of these copies are mutated, the mutation is that the protein is no longer functional. Proto-oncogenes, when they become mutated, are not typically non-functional. They're typically overactive.